ever wonder how drugs really work? Like what's actually happening at the cellular level when you take a medication or maybe use cannabis?
It's a fascinating question.
It is, right. And today, we're going to deep dive into that world, looking specifically at membrane-bound receptors.
Oh, very cool.
Yeah, and focusing on the interplay between cannabinoid and serotonin receptors. We'll also be exploring these things called membrane rafts,
Ah, yes.
which honestly sound kind of like something out of a fantasy
A
novel.
little bit, yeah.
But they're actually crucial to how these receptors actually function. And then we'll also be talking about these receptor complexes called homodimers and heterodimers.
Right, so these are receptor tag teams.
Exactly. And they can really shake things up in the body. By the end of this deep dive, you'll have a whole new understanding of what's happening at the cutting edge of pharmacology and what it means for the future of drug development.
Exciting stuff.
Okay, so first things first, let's try to visualize the cell membrane. Help us paint a picture.
Okay, so picture a bustling city, but instead of buildings, you have this dynamic, fluid structure made of fats and proteins.
Oh, interesting.
It's a barrier for sure, but also a major communication hub. It's constantly receiving signals and transmitting information.
I see.
But here's the thing, not everything can just waltz in and out of this city. There are checkpoints, security measures,
Right.
and even specialized zones, kind of like different districts.
Ooh, I like where this is going. Tell me more about these districts.
Okay, so these districts, these are what we call lipid rafts.
Okay.
They're like little islands within the membrane, more tightly packed than the surrounding areas.
Interesting.
And they tend to attract certain proteins, including those crucial receptors we're going to talk about.
So why do these lipid rafts actually matter for how the receptors function? Is it all about location?
Yeah, location. It's a big deal. Think about it. If you're a receptor and you're hanging out in a lipid raft, you're mingling with a different crowd than if you were out in the general membrane area.
Right, makes sense.
And this can influence who you interact with and how you respond to different signals.
Fascinating. Okay, so we've got this dynamic membrane. We've got these specialized lipid raft zones.
Right.
Now let's introduce the stars of our show, the receptors themselves. We're focusing on cannabinoid receptors,
Yes.
CB1 and CB2,
Uh-huh.
and the serotonin 5-HT2A receptor. Why are these receptors so important?
Well, they're key players in so many different bodily functions.
Yeah.
And they're all membrane bound, which is crucial for how they work. For example, take CB1 receptors. They're mostly found in the brain and nervous system.
Right.
And these are the ones responsible for the psychoactive effects of cannabis.
Oh, wow.
But they're also involved in things like pain perception, memory, even appetite.
Wait, so that's why cannabis can give you the munchies?
Yeah, it's all connected. It's messing with your CB1 receptors.
That's wild.
Now, CB2 receptors, those are more common in the immune system.
Oh.
They help regulate inflammation and pain, which is why there's a lot of interest in their potential for treating things like arthritis.
That makes sense.
Yeah.
And what about the serotonin 5-HT2A receptor? I know it's involved in mood, but I'm guessing there's more to it than that.
Oh, absolutely. So much more. This receptor is like a multi-talented artist. It's involved in mood regulation, yes, but also perception, sleep, and, you guessed it, even appetite. In fact, psychedelic drugs like LSD, they target this receptor,
Interesting.
which might explain why they cause such profound shifts in thinking and perception.
Okay, I'm starting to see how these receptors are involved in some pretty important stuff, but
Right.
here's a question that's been bugging me. Why do they have to be stuck in the membrane? Couldn't they just float around and do their thing anywhere?
You know, that's a great question, and it really gets to the heart of why their location is so important. You see, these receptors are the cell's communication interface with the outside world.
Okay.
They're like antennas, positioned perfectly to pick up signals from molecules that can't just cross the membrane on their own.
So they're like the Border Patrol, checking passports and deciding who gets in.
Exactly. And by being embedded in the membrane, they can transmit those signals inward, triggering a cascade of events inside the cell, kind of like sending a message down a chain of command.
Okay, so not just receiving the signal, but also translating it in a way for the cell to understand.
Exactly.
Okay. Now, thinking back to those lipid rafts we talked about earlier, it makes me wonder how much a receptor's exact location within the membrane actually affects its function.
It's a brilliant observation. You're absolutely right. So the membrane isn't just a static wall. It's this dynamic, fluid environment.
Right.
So a receptor's location, especially if it's nestled in a lipid raft, can significantly impact how it interacts with other molecules and how it ultimately functions.
Wow, so even tiny shifts in position could change a receptor's behavior.
Absolutely. It's all about location, location, location.
Yeah.
And to make things even more complex, these receptors, they sometimes like to team up, forming what we call dimers.
Effector buddies.
Exactly. It's like two receptors linked together.
Now I'm really intrigued. Tell me more about these dimers.
Okay, well, there are two main types. You have homodimers, where two of the same receptor type join forces. And then you have heterodimers, where two different receptor types pair up. For example, CB1 receptors, they're known to form homodimers, which can actually alter their sensitivity to cannabinoids.
So two CB1 receptors working together might react differently to, say, THC than a solo CB1 receptor?
Exactly. And this can have a huge impact on how cannabis affects different people. It's not just about the dose, but also how these receptors are interacting.
Wow. This is really making me rethink everything I thought I knew about cannabis. Okay, so we've got this picture of a dynamic membrane, we've got these specialized lipid rafts, and receptors are moving around, sometimes forming these dimer tag teams. It's a lot to take in. But I have to ask, what does it all mean? Why should we care about all these molecular details?
That's the million-dollar question. And it brings us to the heart of drug development. You see, understanding where a receptor is located, whether it's in a lipid raft and if it's part of a dimer, can all affect how a drug interacts with it.
I see. So it's not just about finding a drug that binds to a specific receptor.
Right.
It's about understanding the whole context, the environment in which that receptor exists.
You got it. It's like trying to solve a puzzle. You need to understand how all the pieces fit together. And that's where our deep dive into signaling pathways comes in.
Oh, signaling pathways. Now we're getting to the good stuff. Can't wait to unpack that in part 2.
Welcome back. Ready to unravel those signaling pathways?
Absolutely.
Yeah.
Let's dive into this intricate world of cellular communication.
All right, so you know those receptors we were talking about?
Yeah.
Well, they're kind of like first responders,
Right.
receiving signals from the outside. But the message, it needs to be relayed inside the cell
Right.
for anything to actually happen. And that's where signaling pathways come in. They're like a chain of command, passing the message along.
Okay, I'm visualizing a relay race
Yes.
with each runner passing the baton.
Exactly. Perfect analogy. Now, each receptor triggers a specific pathway, leading to different effects.
Yeah.
And things can get pretty complex,
Yeah.
especially when you factor in those dimers we talked about.
Right, okay, I have to admit, this is where I start to feel a bit lost.
Yeah.
Can we break down a specific example to make it a little more concrete?
Absolutely. Let's stick with our star player, the CB1 receptor.
Okay.
When it binds to a cannabinoid like THC, it activates a specific protein called a G protein,
Okay.
which acts like a messenger inside the cell. And this particular G protein, called G, is like a signal dampener.
So G is like a volume knob turning things down.
Exactly. It inhibits an enzyme called adenyl cyclase, which is responsible for producing another important signaling molecule called CAMP.
Okay.
So by inhibiting adenyl cyclase, the CB1 receptor essentially lowers CAMP levels inside the cell.
Got it. So less CAMP means what? What does that actually do in the cell?
Well, CAMP is involved in all sorts of cellular processes, including the release of neurotransmitters, those chemical messengers that allow brain cells to communicate. So by lowering CAMP weak levels, CB1 activation can indirectly reduce neurotransmitter release.
Okay, so that's how cannabis can affect things like memory and movement, by messing with neurotransmitter release in the brain.
That's a big part of it, yeah. But it's not the only way CB1 receptors exert their influence. They can also directly interact with ion channels, which are like tiny gateways controlling the flow of charged particles in and out of cells.
Interesting.
And this can further fine-tune neuronal activity.
Wow, so it's a multi-pronged approach.
Yeah.
One receptor, multiple ways of influencing the cell. What about the other receptors we talked about? Do they have their own unique signaling pathways?
Absolutely. For instance, the serotonin 5-HT2A receptor, that one activates a different type of G protein called GQ.
So GQ is like a different messenger taking a different route?
Precisely. Instead of inhibiting adenyl cyclase, GQ activates an enzyme called phospholipase C.
Oh, okay.
This sets off a cascade of events, ultimately leading to the release of calcium ions from storage compartments inside the cell.
Calcium ions, those are important for muscle contraction, right?
Among many other things, yeah. Calcium acts as a powerful signaling molecule inside cells, triggering a whole host of responses.
So depending on the receptor and the G protein it activates, you get a different chain reaction, ultimately leading to distinct effects in the cell. It's like a complex choreography.
I like that.
Each receptor is calling its own set of moves.
That's a fantastic way to put it. But it gets even more intricate when you factor in those receptor dimers we talked about.
Right. those receptor tag teams.
Yeah.
How do they affect these signaling pathways?
Well, dimerization can change the game completely. It's like having two dancers instead of one. The steps are going to be different.
Right.
For example, when two CB1 receptors pair up as a homodimer, they can actually change their sensitivity to cannabinoids.
So the same dose of THC might have a different effect depending on whether CB1 receptors are flying solo or hanging out as a duo.
Exactly. And that's just one example. Research is showing that receptor dimerization can have all sorts of effects on signaling pathways, sometimes amplifying the signal,
Wow.
sometimes dampening it, and sometimes even triggering completely new pathways.
This is making my head spin a bit.
Yeah.
But it's incredible to think about all this complexity happening at the microscopic level. So we've got these receptors embedded in the membrane, sometimes forming dimers, and they're all activating these intricate signaling pathways. But what's the point of all this? What are the functional implications of all this molecular mayhem?
Well, it allows for incredibly precise control over cellular processes. Think of it like a sophisticated control panel
Okay.
with multiple dials and switches that can fine-tune everything from neuronal activity to immune responses.
Hmm.
And this fine-tuning, this is essential for maintaining balance in the body, what we call homeostasis,
Right.
and adapting to ever-changing conditions.
Okay. That makes sense.
Mhm.
But I still can't help but think about the implications for drug development.
Of course, yeah.
If we understand these signaling pathways, can we design drugs that target them more precisely?
That's the holy grail of pharmacology. Instead of just activating or blocking a receptor, we could potentially target specific pathways downstream,
Okay.
leading to more targeted effects with fewer side effects.
So we could fine-tune the signal instead of just blasting it or shutting it down completely.
Exactly. And that's what's so exciting about this research. It's opening up a whole new era of drug discovery, one that's focused on precision and personalized medicine.
That personalized medicine, that sounds like something out of a sci-fi movie. Tell me more about that in part 3.
Okay, so we're back, and I'm ready to hear all about this personalized medicine revolution you teased at the end of part 2. It all sounds very futuristic.
It does, doesn't it?
Yeah.
But it's closer than you might think, and it all comes back to understanding those intricate signaling pathways we've been discussing.
All right, okay. Remind me again. How does knowing about these pathways actually translate into personalized medicine?
Okay, so imagine this. You go to the doctor, and instead of just prescribing a standard dose of medication, they analyze your DNA
Okay.
to determine your unique receptor variations and signaling pathway preferences. They might even look at which receptors are forming dimers in your cells.
So it's like getting a personalized blueprint of my cellular communication network.
Exactly. And with that knowledge, doctors could predict how you might respond to different drugs,
Hmm.
what side effects you might experience, and even tailor the dosage to your specific needs.
Wow, that's pretty incredible. So no more one-size-fits-all approach to medication. Not necessarily. I mean, some conditions might still benefit from standard treatments.
Right.
But for many others, especially complex conditions like chronic pain or mental health disorders, personalized medicine could be a real game changer.
Yeah, I see the potential there.
Huh.
But how are scientists actually studying these receptor interactions in such detail? I mean, it all seems so tiny and complex.
Well, that's where the really cool technology comes in. There are these incredible imaging techniques, like super resolution microscopy, that allow us to visualize these molecules with astonishing precision.
Wow.
We can actually see how they move within the membrane, how they interact with each other, and how they cluster in those lipid rafts we talked about.
So it's like watching a microscopic ballet, seeing all these interactions unfold in real time.
It's mesmerizing. And it's giving us unprecedented insights into the dynamics of receptor signaling.
Yeah.
We're no longer just inferring what's happening, we're actually observing it.
That is really amazing. So we're getting this detailed picture of how receptors behave.
Right.
And we're starting to understand how that translates into individual responses to medication.
Yeah.
What's next? How does this knowledge lead to new therapies?
Well, one exciting avenue is the development of drugs that target specific receptor complexes. For example, if we know that a certain disease is associated with the formation of a particular heterodimer,
Yeah.
we can design drugs that specifically disrupt that interaction.
So it's like designing a key that only fits that specific receptor pairing.
Exactly. That's a great way to put it. And this targeted approach could lead to incredibly precise treatments with fewer side effects since you're only interfering with that specific problematic interaction.
This is really making me think about the potential for treating conditions that have been really difficult to manage,
Yeah,
like chronic pain
right.
or neurodegenerative diseases.
Hmm.
If we can understand how those receptors are interacting and what's going wrong in the signaling pathways, maybe we can finally develop effective therapies.
I'm right there with you. There's so much potential here.
Yeah.
And the research is moving at an incredible pace. It's a really exciting time to be in this field.
Well, I have to say, this deep dive has been an eye-opening experience. I had no idea how much complexity existed at the molecular level and how understanding those tiny details could have such profound implications for human health.
Well, it's been a pleasure sharing this journey with you. And remember, we've only just scratched the surface. There's still so much we don't know.
Right.
But with every discovery, we move closer to unlocking new and innovative therapies for a wide range of conditions.
I can't wait to see what the future holds for this field. And listeners, keep those brains buzzing. There's always more to explore in this fascinating world of science.